Okay, so I want to start out by talking about what bioinformatics is, and actually begin with a let me just say bioinformatics and computational molecular biology. These are two terms that are somewhat interchangeable. It's generally concerned with the use of computation, the use of computers, the use of databases in biology. It used to say molecular biology. Now it's even broader than that, but it did really begin in a sense and began in the study of protein before that, and then molecular biology. But our ability to obtain massive biological data, categorize it, organize it, cross-tabulate it, and so on, and the value that people believe can be extracted from that data leads to the need for techniques involving computer science, involving statistics, involving disciplines that weren't traditionally part or traditionally aligned with biology, but now are. And so the whole area of computational biology and bioinformatics has emerged out of that synthesis. And what this class is attempting to do is to teach why we do bioinformatics. There's a little bit on the biology side, how to do some of the bioinformatics, that's in the labs principally, and the ideas that are behind the methods. I'm an ideas person. I don't actually use a lot of these tools, and I understand the ideas much better than I know the particular flavor of the month website. In fact, we'll see some of the course notes that were developed last year or the year before where the web presentation has changed. We have pictures in our notes that show the web looking in some particular way, and that's different. And those things change very rapidly. But the ideas behind the bioinformatics tools actually go back, I could say, in some cases, 50 years. There are ideas that are absolutely critical today in bioinformatics tools that were developed 50 years ago, and except for minor perturbations or glitz that have been added on top of it, really are the same today as they were 50 years ago. The difference is 50 years ago, 40 years ago, 20 years ago, you were kind of an oddity if you studied those things. Nobody really cared. It's the emergence of genomics, the acquisition of massive data, principally sequence data so far, that has made those tools that existed for quite some time, it's made those tools valuable and is the reason that we're all in this course now, or there are courses like this. Okay. So the ability to augment and leverage traditional biological information with computation is sort of at the heart of bioinformatics, and bioinformatics involves the intersection of, I guess this is the time to draw one of these circle pictures. Here's biology, various branches of biology, and then we have computer science, and then we have mathematics, and statistics. And somewhere in the intersection of most of these, it might spill out a little bit, that's where bioinformatics is. And so this picture actually explains the prerequisites for this course. And the prerequisites are generally taken quite seriously. You have to have a certain amount of background in biology. Well, okay, there are some people who have come to me and asked me if that is really necessary. It will be more challenging if you don't. And there are, through our website, there's a section called references or reference materials. There are a couple of papers on there that help people who have a strong background, let's say in mathematics or computer science, and so therefore will have some extra time, but don't have the right background in biology. Those readings will help you learn some basic biology. There's one called Molecular Biology for Computer Scientists, and another one, a primer on molecular genetics. So that's about 10 years old at this point. It's still useful to look at it. In the primer for molecular genetics, anything that they say about specific technologies you can ignore, because those things have long since changed. You can learn in there all about RIFLIPs, which nobody says anymore. It was a nice word. While it lasted. But on the basic issues of molecular biology, that's a good primer to look at. We also have prerequisites, math and statistics. And actually, probability, as opposed to statistics, is much more important in bioinformatics. And so I expect everybody to have had a basic course in probability and statistics, know about permutations and combinations, what a random variable is, what the mean of a random variable is, expected value. We probably won't talk too much about variance, but elementary things like that. The linearity of expectation is a tool that we'll use often. What do I mean by that? Well, I'm getting a little ahead of myself, but let me just say the expected value of the sum of two random variables is the expected value of the first one plus the expected value of the second. We'll see how that comes up. But if this is totally foreign to you, you've never even seen this, and you did take a course in probability, well, you actually did see it. You just have forgotten. Go back and review it. So review elementary probability, and because this will be quite important. Computer science. It's expected that everybody here has had some experience with programming. We're going to use the particular language Perl. And again, one of the things that's available at this website is a first set of notes. There'll be a second set as well. And in that first set, there's an explanation of why we use Perl in bioinformatics. In fact, why other people use Perl in bioinformatics as well. So in the end, you won't become expert Perl programmers. You won't become professional programmers coming out of this course. But hopefully you'll understand how to do some fairly simple things in Perl and have an appreciation for it and why we use it so that you can then learn more as you need it. But if you've never had any programming whatsoever, you may find this to be a struggle because we're not going to really talk about elementary issues in programming. We're just going to be talking about the specifics of Perl. Okay. Some people can just pick it up anyway. I mean, just like isn't any of these materials, smart people can get by even without having had those courses. But those are the expected prerequisites and the useful prerequisites for this class. Okay. So actually, I'm trying to use this thing more than I did last year. So if you can focus on that. Can everybody read this and see it? It goes, last year I used an overhead, and then the camera took that, but it was ended up being kind of fuzzy on the tape. If it goes through here, it'll be clearer on the tape, although it's very difficult to focus up here. But at any rate, this is just a nice quote I like. There's a program called Life Sciences Informatics Program, which is at UC. It used to be at Davis, but it's moved to Berkeley. And some of the basic literature that they put out has this quote. And first of all, the capability to acquire sequence information has become so powerful that the volume of new data generated is growing exponentially. Well, we all know that. This was actually written several years ago where that was news. But the next one is what I think is important. There's a general agreement that significant new understanding of basic biology and biomedical concerns will come from analyzing the vast amounts of data being generated. That's what this course is about. That's what bioinformatics is about. And the ideas for that analysis is really what this class is about. And so important has this domain become that it's acquired its own name, bioinformatics. Okay? So it's the ability, it's the fact that massive amounts of data are being acquired and the need to analyze it, and the belief that by analyzing it, you actually learn something interesting about biology. That was not an accepted belief. It was, in a sense, an open question until recently, and may even still be an open question in some people's minds. And I'm going to talk about that a little bit more in a minute. All right. So we're actually going to focus on sequence analysis in this class. Bioinformatics really has to do with the analysis of the massive amounts of data that are coming into biology. But sequence data, DNA sequence, protein sequence, RNA sequence. Sequence data is the most available, and analysis of sequence data is the most mature illustration of the whole general paradigm of bioinformatics and extracting value from that massive amount of data. So we'll be focusing principally on sequence data, but you should know that bioinformatics, computational biology, genomics, has much more, is much more than just sequence data and sequence analysis. The data that's being developed now involves three-dimensional protein structures, involves expression data that comes from RNA, proposals to monitoring of RNA, proposals to monitor protein, actual protein, amounts of protein in a cell, where it's being expressed, where it's being transported, lots of phenomics, which is phenomes, phenotype, all these new omics, as I had a list of these here somewhere. Almost anything in biology that you want to study, you add an omics, the word O-M-I-C-S to it, and it means the large-scale development of data or large-scale acquisition of data concerning that issue. Protein-protein interaction, regulatory sequences. Again, almost any issue in biology where there's a large-scale, high-throughput, to some extent automated acquisition of that data is becoming an omic and is becoming part of bioinformatics. So generally we call this stuff genomics. That comes from the word gene, but genomics is much more than just the study of acquisition of gene data or sequence data. But the sequence data endeavor is the most mature part of bioinformatics, and that's what we'll focus on. So this leads in really to what I want to say about my completely outside view of biology. I'm not a biologist, so what I'm going to say is intended to be completely foolish and maybe offensive. But this is my view of where biology is today and with respect to bioinformatics. So you can have this large picture. So biology has gone through several different modalities. Actually, I would say the beginning was sort of observation-driven. These are naturalists, went out and collected lots of things, even before microscopes, bug collections and so on, just as sort of an inventory of what's out there. And, of course, a lot was learned from that, but that's, in fact, maybe some people's old view of what biologists do. In fact, the kind of people who do these massive observations out in nature, anyway, are pretty rare these days. Most universities, Davis included, used to have large collections of things that were collected and observed in nature, and people who kept those up and cataloged them, made sure that the ether was fresh or whatever whatever it was. Mostly those people are gone, and there are large collections in a number of places, and Davis is one, that are not really being kept up. But that's observation-driven biology is one modality that has existed in the past. And then another modality, and this is not necessarily intended to be chronological, theory-driven, where somebody has various ideas to try to explain the observations and develops those theories even more to try to explain those observations and the observations that are here. But it's still, in a sense, a theoretical endeavor. So the major modality which has existed in this, for most of this century, or not this century, the previous century, was called hypothesis and experiment. driven, where you have some idea, maybe it comes from theory, maybe it comes certainly from some observations, things that you know about, but you really have some suggested idea of how things work or why things work, and you try to design an experiment to see whether you're right or to learn something more about the various things you're studying and the ideas you have about them. And of course, these things interact. The hypothesis leads you to the experiment. The experiment is, in a sense, an observation. It affects the theory. But this was the major modality of the last part of the 20th century. And the fourth modality is large-scale molecular data accumulation. So you no longer necessarily have a hypothesis, you just have a belief that if you were to find all of the genes in a particular genome, and you could do that in some kind of efficient but exhaustive way, that would be of great value. In a sense, when people were doing hypothesis, experiment-driven biology, often you wanted to find out what was the sequence of a particular gene, or you wanted to locate where that particular gene was. Let's say if you had some phenotype, you have some trait that you know about, and you're trying to find where is a gene that affects that trait, you would do various experiments to try to find that particular gene. Here, we're just going to say, let's find all the genes and all the promoters and all of the enhancers, whatever, in the genome. Let's find them all in some kind of large-scale, high-throughput way, because if it was valuable to find them one at a time, it must be valuable to have all of them. Okay? Now, in a way, this large-scale molecular data accumulation is really similar to this observation-driven biology. In a way, we're kind of back to the beginning. It's just that it's no longer in nature. It's no longer going out and collecting butterflies and beetles. It's in the lab, and there are experiments that you have to do, of course, to gather this data, but it's no longer driven by hypothesis. Okay, so this is really what genomics is about. And when you have this high-throughput, large-scale accumulation of data, then you have to organize it. You have to massage it. You have to make sure it didn't get corrupted in various ways that it could. And then you have to learn something from it. And that's what leads us to bioinformatics. And the point I made about 10 minutes ago is that the ideas, the methods in bioinformatics predate genomics. But it's because biology has moved on to this fourth modality that bioinformatics has become important. It predates genomics, but genomics makes bioinformatics important. So there are a small group of people who were doing things related to bioinformatics well before genomics. It was a nice, quiet place. People left you alone. Nobody really cared that much about what you were doing. And all of a sudden, or people did this as a hobby. I was one of them. And all of a sudden, late 1980s, the Human Genome Project came along. It's like, somebody lifted that rock, and we're one of those little squirrely things, wiggling things underneath there. The light got shined, and all of a sudden, people cared about what we were doing. And then the 6 of us or 10 of us who used to work in that field got joined by thousands of others. So the whole thing has totally changed. But I wanted to give you some kind of historical perspective here on where I think we are and how bioinformatics fits into that general framework. So we're going to be focusing in this class on the methods. That means the ideas. We'll have two parts to this class. This class is titled Theory and Practice. Okay? The theory part is what we're going to do principally in lecture and through the course notes that I'll distribute to you. The practice part is principally what we do in lab because people ultimately want something that's of use. And you'll see in lab a representative illustration of, well, we only have 9 or 10 labs, but you'll see basically one example of several of the main kinds of things that people do through computation in bioinformatics. Each lab will focus on a slightly different topic and a slightly different software utility that's available out there. We'll do the Perl for 2 reasons. One is so that you can learn enough about Perl to be of some use to yourself and to others through programming. And the second is so that through toy illustrations, we can explain the ideas behind some of the methods. So how many people here have used BLAST? Okay, maybe half the people here. How many people have never heard of it? And enough and who are willing to admit it. So BLAST is the principal utility that's used today in bioinformatics. In fact, I should have brought in a quote. Actually, there is, no, didn't bring the book. There's a quote in another book that I didn't bring, which basically said, BLAST is bioinformatics. I mean, I think that's wrong, absolutely wrong, but that was a quote in a particular book, and some people look at it that way. Well, we won't, BLAST is a massive, massive program. We'll use it extensively, but we won't rewrite it. I know people who who just started from scratch and said, I don't like it the way it is. I'm just going to write it myself. We won't do that, and I don't think coming out of this class you'll have the ability to do that. But we will do some toy examples where you program in Perl various aspects of the BLAST program so that you begin to understand better how it works and why it does some of the things that it does. Okay. So this is an introduction both to the field and into the course. Now, I want to put up another sort of high-level view. Today is sort of high-level discussion. Those of you who came to lab are already immersed into some very specific things. You're learning how to use Unix and VI and connecting to NCBI and Entree and starting your Perl exercises. So you've got the concrete part. And so because of that, I get at least 1 or 1.5 lectures where I get to do general hand-wavy kinds of things before we get to concrete stuff in lecture. But, so here's another one. What I care about in this class and what I'm trying to get across is that there's a chain of reasoning that goes between the observations or the corpus of biological data that forms people's knowledge about biology and the questions they have about biology. It goes from that through a number of stages that I'm going to write down to finally something you see in front of you on a screen. So it comes as a surprise to many people that when you call up some web service and there's something on the screen in front of you, that there's actually something behind it that generated it. And by that, I don't mean a photon tube that's generating the image. I mean a whole series of ideas that people thought of, experimented with, got wrong, fixed maybe, and so on, that generated what's sitting in front of you. And we want to see that whole chain of reasoning. And in particular, we want to see what compromises people made along the way and why they made those compromises, why they made those decisions, why they made those simplifications, and whether that compromises, in a different sense of the word, the whole endeavor. When you finally get that tool in front of you, which says, enter your sequence and push this button, and then you get back something that says this sequence is that, or here's something we think is relevant, is it likely they got that right? or if you know what was the chain of reasoning that led to that, you'll know something about how to interpret it or to fix those results. So what are the components of this chain of reasoning? And I'm going to refer to this throughout the quarter and really point out, well, this is something that's happening at this point in the chain of reasoning. And here's something that is at some other point. Well, of course, originally we have data, observations, questions. Okay, this is the corpus of biological knowledge. Some of that's very raw. This may just exist in some traces on a glass slide or in test tubes or in somebody's freezer. Or it may be in a massive number of papers that exist in the literature. But at any rate, there's some kind of data, knowledge that's out there. And as, of course, people accumulate additional knowledge as they do additional experiments, or in this fourth modality, develop more general observations. And then from that comes some biological simplifications or rules. Okay? You just can't have an encyclopedia of data and consider that to be satisfying or a real advance. I mean, a lot of biology is just observational or is just factual or is a lot of particular specifics on how something works. And that, to some extent, is unavoidable. But you don't want to just end there. And I heard a lecture recently by a famous biologist. I won't try to pronounce his name. It's unpronounceable. But who basically was saying most of the data that you learn, most of the specific information you learn about cell biology, molecular biology, genetics, and so on, is just happenstance. It's just the way things work without any particular reason or tremendous value for knowing all that detail. That doesn't mean you don't learn that, because you have to. That's the stock and trade of what you do. But if ultimately you don't get to some simplifications or some rules, something that comes out of that, other than just a huge listing of data, there's very limited progress. So I would like somebody to tell me some biosimplifications. Whether they're right or wrong, you wouldn't be able to have an education here if you weren't taught a few. Anybody know what I'm talking about? When you take a molecular biology course, there is one in particular that is usually highlighted. It's given a name. It's probably a whole lecture devoted to it. Central dogma, right. Okay. And okay. So what is the central dogma? Yeah. Okay, what about DNA to RNA to protein? Okay, so information, I don't know what kind of information, genetic information, molecular information, protein construction information. Anyway, information goes from DNA to RNA to protein. And there's a second part of it. What's the second part of the central dogma?
It's
The direction is unique. It doesn't go backwards. The information flows from DNA to RNA to protein. Now, this is the central dogma as I don't know whether it's 1950s or 1960s. This is the original one you'll find in the old textbooks. But you can see that this comes out of data that was observed, information that was known. It came out of people's minds. Maybe it came because it was pretty. It came before there was really maybe enough information to say that that's right. And today, do we actually believe this? Any part of this no longer seem absolutely right? Yeah. Yeah, definitely. There's one direction that we know is not absolute. RNA back to DNA is not only known, but critical, common, and more functions are being understood over time for this particular direction. What about the other way? Protein to RNA or protein to DNA? I think that's still pretty stable. However, there is information flow from protein to protein, or at least believed to be. Nobel Prize was given for that. Anybody know what that's called? Prions. Okay, so what's my point here? My point is that we have simplification rules. Without them, we just have a mass of data, and you really can't operate that way. But the rules may be right, or they may be 99% right, or they may be right at the moment that you're looking at them for the relevance, for the purposes that you have, or they may be wrong in some minor way, or they may be wrong in some major way. And ultimately, when we have a tool, when we have a bioinformatics idea or tool that's built on here, it may be affected by whether this is right or wrong. And I can tell you that most of the people who build tools in computer science, let's say, tools for bioinformatics, they never look in here. I've never set foot in a laboratory. Well, I've set foot in a laboratory, literally. That's about it. I've never so much as mixed anything in test tubes since high school. So I don't get immersed in data, in real data here. My knowledge of biology comes from this level of understanding. And I have at least learned that any rule that you'll learn has exceptions and may even be, in a very important way, wrong. Anybody have any other biosimplification rule you want to propose? There are probably, if you put your mind to it, at least 30 or so that you could write down. Here's one. One gene, one protein. So central, it almost is at the heart of a definition of what a gene is. I mean, what's your basic definition of a gene? Well, as a geneticist, you might have a slightly different definition of a gene that really relates to heredity, information that's passed across generations. But from a molecular biology standpoint, a gene is a sequence, is an interval in DNA that ultimately specifies a protein, for the most part. There are certain things that are expressed other than proteins, RNA, for example. But at any rate, one gene, one protein was a kind of mantra that existed for a long time. And as I said, it almost is at the heart of what the definition of a gene is. But what do we think about this one now? Anybody have any ideas? Yeah.
It's not true because in complex genetic diseases, interactions between genes might cause different proteins to be made.
Yeah, I mean, originally meaning about 2 years ago, back in the last century, it was estimated there should be about 100,000 genes in the human genome. because there were about 100,000 proteins that people could detect. Some people thought it was as high as 200,000. Okay? Now that the genome sequence has been more or less done, the estimates are closer to 35,000. Well, what does that mean? It's not that 65,000 proteins disappeared. Okay, they're still there. It's that the whole definition of what a gene is has changed because of the realization, and of course it was known before 2 years ago, but not the scale. It's known that the same interval of DNA can in fact specify and code for more than a single protein. Okay, depending on which portions of that transcript are spliced out, principally through a mechanism called alternative splicing. And if you read about 5 years ago, people thought, well, maybe 5% of the genes in the human genome are involved in alternative splicing. You look 10 years ago, it was something weird and wacky that somebody discovered once. But today, people think, well, maybe 50% of all what we call genes have alternative splicing. And in fact, it's very important and happens all the time. Okay, so here's another one of those biosimplifications that you need and you use, but may not stand the test of time. I don't mean to be negative here and say that everything you learn is going to be wrong, but it's really important to understand that we have to use these simplifications. On the other hand, it may be wrong. Now, one of the papers that I've put on the reference list is a wonderful paper by Joshua Lederberg, famous Nobel Prize-winning molecular biologist. It's called the Anti-Expert System. And in it, he lists 10 of these dogmas or these rules that have subsequently turned out to be generally disbelieved. Okay? And he also ends with a prediction about one, which is that protein sequence, okay, here's a general dogma, or a general rule that is common today. The sequence of a protein should imply its structure, and its structure is how it folds in 3 dimensions, and its structure should imply its function. Okay? That's the general belief today, and it's slightly modified by these things called chaperones, but actually some people take the evidence of chaperones. Those are molecules that help or seem to be involved in getting the protein to fold properly. Some people take that as evidence, as even greater evidence for the view that sequence implies structure, and their view is that the chaperone simply provides an environment where the sequence data can then get the protein to fold properly, and not a contradiction to that general view. At any rate, in Lederberg's, his end of this little article is that he predicts that dogma to be overturned at some point. Okay, so let me list one more that's really critical. All of these things actually have a role, very central in almost any kind of bioinformatics tools. But let me list one more of these that is very central in bioinformatics tools, so central that most people don't even think about it. Just take it as absolute, so obvious, how could it be any way else? And that is that functional domains in protein are the most conserved, are more conserved in evolution than are non-functional domains. Well, first of all, what's a functional domain? Anybody want to offer a definition?
Part of the protein that gives a function.
Yeah, it's part of the protein that actually does something important. I mean, this actually, the knowledge that there are such things as functional domains also, which implies that there are portions of the protein which are not that critical, portions that you can play around with and change without changing the function or the three-dimensional structure that much. That, by the way, is overturning of a dogma. I have a wonderful Scientific American article from the 1960s about protein evolution and protein structure by a very famous person of that time, I won't name, which basically said every amino acid was gold. You change any amino acid in the protein, and you're going to mess it up. Okay? Well, now we know that's just not generally true. There are specific ones in specific proteins that are extremely important, and you change it and you can mess it up. You can cause all kinds of diseases, even by single mutations of that type. But to a very large extent, many others can be mutated within a wide range of different amino acids without having any real effect. Okay, so today the view is we have functional domains. We have sections that are functional, in fact, functional in a particular protein, and there are some that are not so functional. Okay? And the other part of this is that there is an evolution. There's a change in the proteins over time. And this biosimplification rule says that the functional domains are going to be more conserved throughout evolutionary history than the non-functional domains. In fact, we use this in reverse in bioinformatics to try to find the functional domains. So if you have a whole bunch of proteins that you think do similar or related things, and you have their sequences, and you compare them, and you find, oh, here's a chunk that seems to be highly similar, that's probably functional. That's probably a domain. That's probably important. Okay? So this is so ingrained that we use it in reverse as a strategy for finding functional domains. It's so ingrained that people rarely even say it explicitly and almost never question it. Anybody want to suggest why this might be true? Why do people believe this?
Because of evolution.
Because what?
Because of evolution. Because if the function has changed, then there's totally difference in chance of survival. But if the function remains same, then there's not much. Even the sequence have changed, there's not much difference in chance of survival. So the one sequence that's important for the functions have to remain in order to.
Okay, the last sentence of what you said is those sequences that are important for function have to be conserved. You're kind of just restating it. The view is, I'll tell you the answer that's so ingrained in us, is that you take something that's important and you make a change in it, you're likely to mess it up. Okay? It's likely that if you take you open the hood on your car and you take two hoses that are there that you don't understand, and you just kind of reverse where one is going and where the other is going, odds are that car isn't going to run too well after you do that. Okay? And that's the view here, that things that actually are important for the operation of the protein, if you change them, it's going to be deleterious. Okay? But I want to point out, and this is believed today and hopefully believed from empirical observation, but before we had empirical observations about proteins, before there was a lot of data, people made very reasonable cases for the exact opposite. And the primary person who made that case was at UC Davis, a guy named Dubchansky, who really believed that it was the non-functional portions of the protein that should remain the most constant. And the functional parts, if you compared different proteins, were the ones that were changing the most rapidly. And it all really comes down to whether you view a typical mutation as being beneficial or deleterious. It comes down to an empirical fact that where people didn't have data back in those days, in the 1930s, but they had theories, they had ideas, and they could make a very reasonable argument for exactly the opposite. Okay? So again, my point here is to point out that there are simplification rules that they're not necessarily always right. They're not always necessarily obvious. You can make arguments in the absence of data for something which is totally opposite to the simplification that we take today. And some of these things change over time. Now, I only gave you one step, or two steps, sorry, in this chain of reasoning. I want to go next time to the next several steps. I have two more. I have 30 seconds according to that book, according to that clock. And I just want to point out some of the books. The one book that's required in this course is a book on Perl by Johnson. It's not in the bookstore yet, apparently. But for the first couple of weeks, anyway, the notes that are on the web should suffice. There's another one called Beginning Perl for Bioinformatics, O'Reilly. I think, actually, my personal view, I've read this now. It came out about 3 or 4 months ago. I think it would be very difficult to learn Perl from this if you didn't already know how to program or if you didn't already know Perl. But once you do know enough of Perl, this has some wonderful examples of using Perl in bioinformatics and developing larger programs. So if you're really serious, eventually go out and get this. Algorithms in Strings, Trees, and Sequences. This is a book I wrote. About half or 3/4 of the lectures are actually taken from this book, and I have, shouldn't say this too loudly, my publisher might hear it. The notes that you have there are mainly taken from here. However, in the end, this is what we're going to do in this course is about 1/4 of this book. So again, if you're really serious, you might eventually get this, but I'm not asking people to do that originally. Biological sequence analysis by Durbin and Eddy. We'll use about 50 pages of this. I won't be able to distribute it to you, but the material will be roughly 50 pages of this book. This is a very serious book. Again, for people who are serious, it's related to probabilistic reasoning. In fact, that's the subtitle, Probabilistic Models of Proteins and Nucleic Acids. It's really about hidden Markov models. If anybody's ever heard of those, we'll discuss them. This is the book to get for that.